Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine this. What if we could get a really powerful Alzheimer's drug right into the brain where it's needed? And not only make it way more effective, but also, get this, almost completely wipe out a major dangerous side effect. That's actually the incredible promise from some very recent science. So today, we're diving into a really fascinating new approach that, honestly, could rewrite the rules for treating diseases like Alzheimer's.
It really could. I mean, the fundamental problem with the anti-amyloid antibodies we've had for Alzheimer's, it's always been sort of two things, right? They really struggle to get across the brain's protective barrier efficiently.
The blood-brain barrier.
Exactly, the BBB.
Yeah.
And even when they do get in, they often come with this serious safety issue called ARIA.
Right, ARIA. We hear a lot about that.
Yeah, amyloid-related imaging abnormalities. You see things like swelling, edema, or even little bleeds on the MRI scans. But this new research, it offers a really elegant solution. It seems to tackle both the delivery problem and the safety problem at the same time.
Okay, so that's our mission for this deep dive then. We're going to unpack this cutting-edge research. It's from the journal Science. We'll explore how this really cleverly engineered antibody, like a transport vehicle, could maybe bring in the next generation of Alzheimer's treatments, offering, better results with way less risk. So let's maybe just set the stage first. For years now, the focus has been clearing out those amyloid beta plaques in the brain, right? That's been the main strategy for Alzheimer's. And antibodies designed to do this, these anti-AT therapies, they have shown some promise in trials. We've seen hints they can slow things down, but there's always been this huge obstacle, the brain's own, bodyguard.
That's right, the blood-brain barrier, the BBB. It's this incredibly selective layer of cells lining the brain's tiny blood vessels, the capillaries.
Yeah.
Its job is crucial, keep out harmful stuff, pathogens, toxins. But the downside is it also blocks most large therapeutic molecules, things like antibodies.
So they just can't get in easily.
Right. Even if a drug is really powerful in the bloodstream, it might just be locked out of the brain, or only tiny amounts get through.
And okay, even for the small amount that does manage to sneak past the BBB, we hit that other really big problem for patients, ARIA. Anyone who's been following the Alzheimer's drug news, lecanemab, donanemab, they know ARIA is a big deal.
Oh, absolutely. ARIA is a serious side effect. Often it's the thing that limits the dose you can give. You see it on the MRI scans, like we said, either brain swelling, that's the edema, or these tiny microhemorrhages. And it seems to be linked to the antibody interacting with amyloid that's built up in the blood vessel walls, causing inflammation right there.
Which is obviously not good.
Definitely not. For doctors, it means lots of MRI monitoring, maybe having to lower the dose, sometimes even stopping the treatment altogether. And that creates huge barriers. For patients getting the treatment, for clinicians prescribing it, it's this major safety versus efficacy trade-off we've desperately needed a solution for.
So, okay, two massive challenges. Getting enough drug in and stopping the bad side effects once it's there. This new research says it can do both. How? What's the core innovation here?
What's really fascinating, I think, is the engineering strategy. It's quite elegant. They've essentially built a targeted transport vehicle right onto the anti-amyloid antibody itself. And this vehicle specifically targets something called the transferrin receptor, or TFR.
TFR, okay.
Yeah, it's a receptor that's found in really high amounts on those endothelial cells that make up the blood-brain barrier. Normally, its job is to ferry essential things, like iron, into the brain. So the idea here is basically to hijack that natural process.
Like getting a ticket on the brain's existing delivery system.
Exactly. It's called receptor-mediated transcytosis. They're using TFR to actively shuttle the antibody across the BBB.
Okay, let's dig into this brain shuttle a bit more. They call it ATVcisLALA, quite a mouthful.
Yes.
How is it engineered to be so smart? Because people have tried using TFR before, right? And ran into problems.
That's the key point. Earlier attempts often had issues. The antibodies bound too tightly to the TFR. So they either get trapped in the barrier cells or they'd cause problems out in the body, like depleting precursors for red blood cells, causing anemia. That was a big one.
Right.
This new design uses what they call asymmetrical FC mutations, the cisLA part. It's really sophisticated protein engineering. These mutations create this perfect sort of sweet spot in how strongly it binds to TFR.
Okay.
Strong enough to efficiently trigger the transport across the barrier, but not so strong that it gets trapped or saturates the system or causes those peripheral side effects. It's about getting the binding and release kinetics just right.
So it's like having a key that unlocks the door perfectly, lets you in, and then comes right back out without jamming the lock. And crucially, once it gets inside the brain, it still needs to do its main job, right? Clearing the amyloid plaques. How do these mutations affect that part?
Precisely. That's the other clever part of the design. These specific mutations, the cisLA ones, they're designed to make the antibody sort of invisible or inert to immune cells while it's in the bloodstream.
Ah, okay. Safer journey.
Exactly. It prevents unwanted immune activation on the way to the brain. But, and this is critical, once it crosses the BDB and gets into the brain tissue, it still has its full therapeutic function. The FC region, the antibody's tail end, is still perfectly capable of binding to FC receptors on microglia.
The brain's own immune cells.
Right. And that binding is the signal that tells microglia, hey, eat this plaque. It triggers phagocytosis, the cleanup process. So it's a beautiful balance. Stealth and safety during transport, but full activity at the target site.
Wow. Okay, so if I'm getting this right, safer in the blood, gets into the brain much better using this TFR shuttle, and then it can effectively trigger the removal of amyloid plaques. That really does sound like a triple win. Designing something that complex in a computer or a test tube is one thing, but, the proof is always in the pudding. How did they actually test this? Did it work in living systems?
Yeah, they started, as you'd expect, with in vitro tests, basically, in lab dishes. And there, they confirmed the basics. ATVcisLALA, this new construct, it didn't cause that TFR-mediated cell toxicity that some older designs did. So check one for safety.
Good.
And crucially, it still triggered the killing of cells when it bound to the ill target. So check two, it still had its intended therapeutic effect. That confirmed the dual design worked on a cellular level.
Okay, makes sense. But the really exciting stuff, for me anyway, is usually the in vivo data, the mouse models. What happened there?
Right. So they moved into mouse models of Alzheimer's. And the results were pretty striking. First, safety. A single intravenous dose of this ATVcisLALA, it showed a major reduction in that circulating reticulocyte loss we talked about.
The red blood cell precursor issue.
Exactly. That was a major hematology problem, a safety flag with older TFR approaches. This design largely avoided it in vivo. Big safety win.
Okay, that's huge.
And then delivery. This is pivotal. It dramatically increased the amount of antibody that actually got into the brain compared to what was in the blood plasma. And importantly, the concentration was higher, specifically in the parts of the brain that had the amyloid plaques.
So it was getting to where it needed to go.
Yes. They even compared it head-to-head with another way of linking an antibody to TFR. And only their ATVcisLALA design managed to boost brain entry without causing the reticulocyte loss. It really highlighted that their specific engineering was superior.
Okay, so they've cracked efficient brain entry and avoided a key peripheral side effect. Now, the million-dollar question. What happened inside the brain regarding ARIA? Did changing the delivery route actually fix the ARIA problem?
This is where it gets really interesting, connecting delivery mechanism to safety. See, conventional anti-amyloid antibodies, they tend to enter the brain mainly through the cerebrospinal fluid, the CSF, and these spaces around the larger blood vessels, the perivascular spaces.
Right.
This means they often end up concentrated around the bigger arteries. And unfortunately, those arteries are often sites where vascular amyloid builds up.
Ah, so the drug goes right to the trouble spots, but in a way that stirs things up.
Precisely. That concentration around arterial amyloid seems to be what triggers the inflammation leading to ARA. It's like pouring fuel on a smoldering fire.
Okay, so the old way uses these arterial highways, which are risky. What route did the brain shuttle take?
ATVcisLALA showed a completely different pattern. It wasn't concentrated around the big arteries. Instead, it achieved this really broad distribution throughout the brain tissue itself, the parenchyma.
Spread out more evenly.
Exactly. Much wider spread, leading to better engagement with the amyloid plaques throughout the brain, not just near the vessels. And the crucial finding from their 3D imaging was that it primarily crossed the blood-brain barrier through the capillaries.
The tiny vessels, not the big arteries.
Right. This capillary-driven delivery, bypassing those inflamed arterial zones, seems to be the absolute key difference.
So taking this different, more diffuse capillary route did it actually prevent ARIA in the mice?
The results were, frankly, remarkable. They use an Alzheimer's mouse model that's known to get pretty bad vascular amyloid, a model where conventional antibodies do cause ARIA-like problems. With ATVcisLALa treatment, they saw a near-complete elimination of those ARIA-like lesions.
Wow, near complete.
Yeah. The MRI scans showed dramatically fewer signs of swelling or microbleeds compared to the mice treated with a conventional-style antibody. It was a night-and-day difference in terms of safety on the scans.
That is huge. I mean, truly huge if it translates. And did they look closer at the tissue level? Did the cleaner MRIs match up with less actual damage in the brain tissue?
They did, yes. The histopathology looking directly at the brain tissue under the microscope, it completely backed up the MRI findings. Treatment with ATVcisLALa led to significantly less inflammation around the blood vessels and the meninges, fewer hemosiderin deposits.
Which are the signs of old microbleeds, right?
Exactly, the iron remnants from tiny bleeds, and fewer microinfarcts, tiny strokes. So it clearly demonstrates that changing the route of entry, going via capillaries, spreading widely, avoiding those arterial hotspots, it really does dampen that whole inflammatory cascade and the vascular damage that we see as ARIA. It really makes you think, doesn't it? Is how a drug gets into the brain maybe just as important or even more important sometimes than just what the drug is? The evidence here is pretty compelling.
It really does feel like more than just, tweaking an existing idea. This feels like a proper step change, a conceptual leap. What does this really mean looking forward for Alzheimer's treatment, maybe even other brain diseases?
I agree. I think this work marks a real maturation in how we design these BBB shuttle systems. The real innovation here isn't just, let's get more drug into the brain. Lots of efforts aim for that. The leap is explicitly linking that enhanced delivery to solving a major clinical safety problem, ARIA. They've engineered the delivery mechanism itself to mitigate the side effect.
So engineering for safety just as much as for efficacy.
Exactly. It's tying the molecular design directly to a critical clinical issue. That's more sophisticated. And the preclinical results showing ARIA mitigation are just much stronger because of it.
So this isn't just, interesting science for scientists. This could genuinely change things for patients and doctors dealing with Alzheimer's.
Oh, absolutely. If this holds up in human trials, and there are early signs from similar approaches that this could fundamentally change the game for amyloid immunotherapy. Imagine being able to effectively separate the benefit, the plaque clearance, from the major risk, the vascular damage. That's the holy grail here.
Yeah.
It could completely reset how clinical trials are designed. Maybe allow for more effective dosing regimens, perhaps clear plaques faster, because you're not constantly hitting that ARIA ceiling. And think about the practical side. Fewer requirements for constant MRI monitoring, fewer patients having to stop treatment because of side effects. That changes the whole equation for patients, doctors, and even for, the payers covering the treatment.
And this clever TFR shuttle idea, this ATVcisLAla approach, is it just for Alzheimer's and anti-amyloid antibodies, or could this be used more broadly?
Oh, this is definitely seen as the platform technology. That's a huge part of the excitement. The basic principle, using engineered TFR binding to shuttle things across to BBB safely via capillaries, it's potentially applicable to many other targets in the brain. Well, think about targeting tau, another protein involved in Alzheimer's and other neurodegenerative diseases, or alpha-synuclein for Parkinson's disease, or even delivering totally different kinds of therapies, maybe things like small interfering RNAs, mRNAs, or even therapeutic enzymes that need to get into the brain to work. It's potentially a universal delivery system for large molecules.
Wow.
And actually, there's already some early validation happening. There's another TFR-enabled antibody, trontinemab, in clinical trials. And the interim data that's been released suggests it does lead to rapid plaque clearance with a significantly lower rate of ARA compared to the first-generation antibodies.
So it's not just theory anymore.
Exactly. It's just this whole concept, smarter delivery equals safer, potentially more effective treatment is starting to bear fruit in humans.
So wrapping this up, what we've really learned today is pretty incredible. By cleverly engineering an antibody to basically hitch a ride on the brain's own built-in transport system, scientists haven't just figured out how to get more drug into the brain. They've also, remarkably, made it much safer just by changing how it gets delivered, avoiding those danger zones.
Yeah, this deep dive really shows, I think, a pivotal shift. Moving therapeutic design beyond just does it hit the target, to really integrating safety and precision delivery right from the start. especially for complex chronic diseases like Alzheimer's. That integrated approach is just paramount. It's about working with the body's biology, using its own systems smartly.
It's such an elegant solution, isn't it? That smart design makes all the difference. So maybe something to think about as you go about your day. If we can engineer medicines to navigate our body's most complex, most protected systems, like the brain, with this kind of precision, what other medical challenges that seem impossible today might we be on the verge of solving in the next, say, 5 or 10 years, just by understanding and cleverly adapting to our own biological systems? The potential is really quite astounding.